All right. Well, hello, everybody. Hope you're doing well today. So what I'd like to do is just a quick walkthrough of this neuron simulation. I was asked to see if I could sort of go through and explain this for anyone who may be gone from class. So first thing I want to do is sort of try and provide some context for what we're looking at here. So sorry for sort of blurry image here, but there are different parts of a neuron. You have the dendrites that receive signals, the cell body which has all of those different organelles, and then here's the axon that sends the signal. What we're going to do is focus in on the axon and then the membrane of the axon. So again, our model is a piece of this axon. So if we sort of look at the model, this is the membrane that's on the axon. And of course, within the membrane are different proteins that are embedded. One is the sodium potassium ion pump. This pump pumps out 3 sodium ions, which are in blue here, and then it pumps in 2 potassium ions, which are yellow. Now, notice that both sodium and potassium have positive charges. So when we operate this pump, if you pump out 3 positive charges and you only replace it with 2 positive charges, then what happens is you create a charge differential. Just like if you're spending $3 of your money, but you're only depositing $2 in the bank, your bank account will go negative. Here, if we're pumping out 3 positive charges, but we're only bringing in 2 positive charges, it's going to create this gradient. It's relatively positive outside the cell and relatively negative inside the cell. All right. Now, there are these different proteins embedded here. The sodium potassium ion pump creates the gradient. Again, it pumps out sodium, pumps in potassium. This leaky potassium channel could allow potassium to leak out, which helps make it more negative in the cell. And then we have a couple of voltage gated channels. The voltage gated sodium channel, usually this gate is closed, so sodium can't move across the membrane. But when stimulated, this gate can open, allowing sodium to pass through. Same goes with this voltage gated potassium channel. A change in voltage can open these gates and allow potassium to flow through the membrane. All right, so let's start to take a look at the packet here. So I'm going to run through the questions. So the first question reads, which direction would sodium ions tend to move if you open the gates? So sodium is more concentrated on the outside. So based on concentration, sodium would flow into the cell. If you think about it in terms of charge, well, remember, like charges repel one another and opposite charges attract. So it's positive outside the cell and sodium has a positive charge. So since like charges repel one another, sodium would tend to flow in. So based on concentration and based on charge, sodium would tend to flow into the cell. So if we open the gate, sodium would flow in, it would pass through the channel here, and then we wind up with some more sodium inside the cell. Let's see. So the next question is, if we look at the number of sodium ions on each side of the membrane now, compared to the cell at rest, are there more sodium ions inside the cell than before or fewer? Well, obviously, if sodium flows in, you're going to have more sodium ions inside the cell than before. So the question becomes, does the internal medium of the cell now become more positive or more negative? Well, if you have all these positively charged sodium ions flowing in, it's going to make it relatively positive inside the cell. So let's switch our Post-it notes. So now, because all the sodium rushed in, it's positive inside the cell and negative outside the cell. So next, let's think about the potassium ions. If we were to open these potassium gates, well, based on concentration, where would potassium go? There's a lot more potassium inside, so it would flow out. Additionally, if we think about charge, well, potassium has a positive charge, right? And if we look, it's now relatively positive inside the cell. So like charges repel, opposite charges attract. So again, if this gate opens, potassium would flow out. So we can imagine potassium ions flowing out. So I'll just sprinkle some potassium ions here. So now potassium is flowed out through this gated channel. When that's the case, there are now fewer potassium ions inside the cell than before because all these positively charged ions flowed out. So now the internal environment of the cell is more negative than it was before. So now it goes back to being more similar to what it was like originally. Okay, so basically what happens is the sodium gates open and the potassium gates open. Sodium rushes in, potassium rushes out, and it causes this spike in electrical activity that sends a signal down the axon. Let's see. Now, a fair question is about what's called the refractory period. With the refractory period, after this gate opens and closes, it stays closed again for a few milliseconds so that when sodium flows in, it can spread out, but it only travels, or it only opens gates farther down the axon. So that helps ensure that the signal travels down the axon. Another thing to consider, if we kept opening and closing these gates, eventually the sodium and potassium ions are going to reach equilibrium. And if you keep opening and closing these gates and the sodium and potassium reach equilibrium, well then you're not going to get any more electrical signal. So with neuronal fatigue, if you keep opening and closing these gates repeatedly, if you keep having action potentials again and again and again, these ions reach equilibrium and the neuron stops firing. So how do we get the gradients reestablished? How do we get sodium back out? How do we get potassium back in? Well, luckily we've got a protein for that. That's the sodium potassium ion pump. So this pump is always working and it's moving sodium out, potassium in to help reestablish these gradients so we can keep sending electrical signals. All right, now one thing to note with this, what we've looked at here is unmyelinated. You can have myelinated axons, and when you myelinate these axons, it obviously covers some of the gates, right? But what that does is it lets the signal jump from one part of the neuron to the next. So it will actually reduce the resistance and speed the transmission of the signal. So myelin actually makes this happen faster. So that's sort of a quick walkthrough of what happens with this simulation. If you have any further questions, let me know. Otherwise, I hope you have a great day. Thank you very much.